Hello.
After that, he went on to a postdoc at Oregon Health and Science. Dr. Rhodes has brought
Test.
patients and has received continuous funding over the years from both NIH and private organizations. He's received a long list of awards and a number of invited talks over the years, and has been responsible for training in numerous amounts of successful Ph.D. and postdoc trainees. So, Dr. Rhodes, thank you very much for accepting your guest lectureship this year with series. Excited to introduce Dr. Rhodes and pass off to him to talk on the origins of exercise.
For the mic on Zoom to work, you gotta use this on or handheld.
Hey, thank you so much for that introduction. And thank you for inviting me. It's a great honor to be here. I met a lot of really cool people today, and I'm especially honored to be here to honor Frank Booth, who obviously is a major figure in the field and someone who I've followed over the years. And I was particularly impressed yesterday to see all the amazing trainees and all the trainees that he's educated, and obviously the love and the community and the family that's there. It's really admirable, and I kind of have a similar kind of philosophy in the way I run my lab. So I was really, really great to see that, and I'm really happy to be here. So I think this is definitely going to be in a Frank Booth style talk. I'm gonna talk about a lot of things that I think that he will find relevant and interesting. Let me see if I can figure out how to move this forward, though. You know, nope. Or this. That's weird.
Yeah, it was weird.
Hold on one sec. Let's see. Escape. No, it's frozen. Oh, there you go. How did you do that?
I don't know. It just froze for a second.
Click. Oh yeah. All right. So we're back. So okay, I was here in 2017, and I gave a talk, and I made this statement at the top here, boldly. If you want to maintain cognitive health throughout your life, the very best thing you can do is exercise on a regular basis. And I know there are a lot of nutritional biologists here, and I also studied nutrition, and absolutely nutrition can affect the brain. But for the phenotypes that I'm interested in, like major changes in cognition, changes in the number of new neurons in the hippocampus, nutrition is a much minor effect than exercise. As I'll talk today, exercise does something very profound to the brain, and I'm super interested in studying and understanding what that is. And I mentioned that it is really good at cognitive and maintaining cognitive performance. And if you think about it, when I talk to students, it's a little counterintuitive, right? Because like exercise, when you're exercising, you're not necessarily like cognitively engaged. And so why is it that exercise actually enhances cognition is a little bit of a mystery. It's not a clear thing. One might think, well, the best way to maintain cognition is to, do crossword puzzles and computer games and challenge your brain. And those things work to some degree, but they're much more specific. So if you do crossword puzzles, you'll be really good at crossword puzzles, but it not necessarily transfer to other tasks. And so the real cool thing about exercise is it doesn't train any one particular task. It's very broad in its effects. And I think I'll touch on a little bit about why that is. And it has a little to do with, I think, its influence on the brain region, the hippocampus, which I'll talk about. But in humans, there's been tons of research over the years. This includes epidemiological studies where they measure the amount of exercise you do and the fitness training you've done, and they show increases in processing speed and all these things. Also randomized controlled studies, some of these done in my university by Art Kramer, you may have heard of, who randomized older adults into those who are exercise training 40 minutes a day, 3 times a week for 6 months, bring them back into the lab and showing changes across the board on many, many different cognitive tasks. Of course, in animals, there's zillions of studies done, some by Frank and others, who have showed that exercise can improve performance across many tasks. There are 2 major research questions that my lab has explored. I had a lot of overlap and interest with Frank over the years. The last 20 years, I basically focused on these 2 main questions. One of them is, how does exercise enhance cognitive performance? And of course, if we can answer that question, that is a massive finding because, as I'll show you, exercise can grow new neurons in the hippocampus. The hippocampus is an area of the brain that atrophies as we age. It atrophies with Alzheimer's disease. It atrophies with depression, anxiety, PTSD, all the things that afflict us, obesity. I know many of you study. So if we can figure out how exercise can enhance cognitive performance and return integrity to the hippocampus, that's a big deal, okay? And there's a lot of people interested in this, and people have been studying this, and we still don't really know the answer, but I'm gonna give you a few clues that I think are kind of cutting edge new ideas in this area. Another area that Frank especially worked closely on, and I as well, is what regulates individual differences in motivation for exercise. If we can figure out how exercise enhances performance, we may be able to develop some medications and treatments for people who don't want to exercise. But we know that we can solve like most of the problems that afflict Americans and Western societies if we could get people to exercise regularly. And so that's a huge topic, much more complicated, it turns out. And I don't think we've really made that much progress understanding at least the genetics and how the genes affect the development of the brain to cause increased motivation. We've made some headway there, but a much more difficult problem. And I'll tell you some things along the lines there as well from what I've learned. But I want to start with the second question, because that's very close to some of the work that Frank has done. What regulates individual differences in motivation to exercise? We all know that some of us like to run. My sister's an exercise addict. I was talking to people. She gets pleasure from running back up and down, Grand Canyon. Whereas, most people don't really like to exercise. And most people are sedentary. What is it about us? Some of this is genetics, okay? And Frank and I have proved this through various different selective breeding and strategies. And so there are genetic differences among us, like there are for anything you can measure, that influence how much we want to or motivated to exercise. But there are also environmental factors, as we know, that can come to play and can shift, even though we were maybe genetically proposed to exercise. If we experience some severe stress or have other things happen, that can actually substantially suppress activity. So what is it doing? How does stress reduce activity? How do genes increase activity? That's something that I've been studying over many years. I started in Ted Garland's lab. This is back in the 90s, and working with this epic selective breeding experiment. It's still one of the most impressive, unprecedented selective breeding experiments in a vertebrate animal, to the extent that it's replicated. There are 4 replicate high running lines and 4 control lines. I'm supposing most people are used to seeing graphs like this, but because Frank has showed graphs like this of selection over the generations. But hopefully each one of these data points represents the average, in this case, wheel running in kilometers per day, of the entire population. There's like 100 animals in this data point, right? So you're looking at a huge amount of data here, 4 control lines, 4 high active lines. And I did my Ph.D. on these mice. And when I joined the lab, Ted developed this model to study how exercise physiological traits would support increased activity, like aerobic capacity, numbers of mitochondria, size of your heart, things like that. And the first couple of years, that's what I was measuring. I was measuring aerobic capacity. I was measuring heart size and muscle size. And we were looking at each other and overdoing the data. We were about around this point, generation 25. And you can see the animals are running like 3 times as much as the control animals. But there were no differences in any of these exercise physiological traits. And I remember the day we were both looking at each other like, maybe this is in the brain, because it's voluntary behavior. And in retrospect, of course it's in the brain. The animals have access to a running wheel. And what differentiates how much animals run, at least at the beginning, is not how strong their muscles are or how much aerobic capacity that is, but it's how much they want to run. Now, I should say that later, after you max out motivation, we have now seen exercise physiological traits change. So it's not that they don't, but it's that they follow, because the first thing that changes is behavior and the neural physiology that motivates the animal to do that. And once you max that out, you can now start seeing changes. I'm not going to talk about the research here because this is old stuff. This is from the 90s. A lot of this is published. I will summarize a little bit about some of the stuff we discovered. But before I do that, I want to talk about a selective breeding experiment that I started in my lab, really in the spirit of Frank and Ted. I understood the power of these experiments, and I really wanted to develop this in my own lab. At the time, I was really interested in developing a model for ADHD. So the wheel running model was more like high activity. And people don't necessarily think of running wheels as being, it's obviously hyperactivity, but it's a particular kind of exercise-like activity. But I wanted to breed mice for high levels of physical activity in their home cages. And so what I did is I set up 8 video cameras in an animal room, and I had this ability to track 64 animals at a time. You're seeing 32 here from one computer at another computer. And I did this, and I selected the animals that moved the most under these conditions for up to 12 generations. And again, I didn't notice, I don't have the replication that Ted has. So we were just doing 2 lines here. But we were able to show that this was obviously heritable. By the end, these mice were running about 1 kilometer per day in their cages. By the way, many of you, when you walk in the animal room at night, you can obviously see which ones are which. The animals that are hyperactive are running around. All the bedding is spread out of their cages. So I should say that these are very powerful models. They prove that genetics can underlie different individual variation in propensity and voluntary desire to move. The million-dollar question, though, is we know that there are genetic genes that underlie this, right? There are genes, physical, structural changes in sequences of DNA underlying this behavior. But what genes and how those genes affect the development of a brain to cause this activity is still pretty much a wide open question. I think the reason for that is that one is very complex. There's like thousands of genes potentially, right? The other one is that I think part of the issue, and this is going to be a theme that's coming up, is that it's very difficult when you think of genetics, genes on one hand, which have very long time scales. Like when you think of how does a gene affect biology? Well, it has to transcribe something and translate something. Or how do genes change? Well, they change over evolutionary history. Okay, well, that's a very long time scale. What about brain cells? How do they work? Well, they work on milliseconds, right? So how do you get something like a gene to affect something like brain activity in milliseconds? Well, we were thinking, oh, it must be the genes are in the neurons and how the neurons are different. Well, possibility is that those genetic changes are maybe affecting some of the support cells, like astrocytes and microglia. And those are positioned, right, to transfer slower timescales into faster timescales. Astrocytes and microglia are touching and feeling and controlling large numbers of neurons, and large numbers of neurons firing is what can control behavior. So I think part of the reason why this has been difficult is because of epistasis, because it's very difficult to understand how multiple genes interact together. And another one is the cell types that we're focusing on. And I think if we switch to thinking about some of the support cells, we may make some better traction in the coming years. I don't want to show you any data from that study because it's old stuff and published. And I want to now switch to unpublished data. So most of the data I'm going to show you from now on are stuff that's not published. And this is one of the studies I wanted to show. Let me just move this outside. This is not a genetic model, but you can see it has like a very similar pattern to it. This is an environmental effect. It's a study that I've been doing with I'll show you a picture. This is with my first graduate student, Peter Clark, who's now a professor at Iowa State. And so he did the experiment, and I'm collaborating with him and another undergrad in my lab from 2007, who's now a professor at Emory, who is basically doing the single nuclei RNA sequencing. I'm going to talk about that in a minute because I really want to sell this method. It's a really, really exciting method. Some of you may have heard of it, some not, and I really want to talk about that later. But let me just say that this is one single bout of inescapable shock. This is the model that's a very common model where they basically restrain the animal and it receives on average around 100 shocks. It's a lot of shocks within about an hour. It's not precise because they're randomly given these shocks. They can't predict when they're going to get them. So it's a pretty severe stressor, but it's one single event, 1 hour or so of stress. And you can see that animals that were not stressed, these are male Sprague-Dawley rats, run characteristically quite high levels, ramping up over several days. It's very characteristic of what you see. Whereas look at what's happened to these animals that were stressed. They're basically not running at all. So this is a major sedentary-like behavior that we're seeing here. And this one I am going to show some data for. I just wonder, so this is a really shitty picture because it's really old, but there's Peter Clark when he was my graduate student, and there's Zach Johnson, an undergrad in 2007. And this is a collaboration now with both of them as PIs. So it's pretty exciting for me. But let me say the experimental design of this, very straightforward. This was all done in Peter Clark's lab. And we are basically analyzing some of the single nuclei data from this experiment. But basically, we have 4 groups of rats. There were 6 rats in each group. Some of the rats were stressed. Half the rats were stressed. Half the rats were not stressed with that paradigm I was saying. And then they were placed with running wheels or not without running wheels. I showed you the running data from this group and this group, right, a minute ago. So we have these 2 groups that we're going to be looking at. The striatum, which I know is a lot of area of brain many of you are interested in because this is an area of the brain that's involved in motivation for voluntary behaviors and particularly voluntary activity. And so we then took the striatum from these rats, isolated the nuclei. So you take a tissue, right? They're all the nuclei. Why nuclei? Why not cells? Well, brain cells are very complex, right? They have these processes, they have the axon, and eventually we're going to need to take these cells and put them in a microfluidic device so that we can so we can profile the gene expression in each cell, each unit, right? And so a neuron, people have done it with cells, but the problem is you tear off all the pieces and who knows what you're looking at. But when you take a nucleus, that's a nice spherical thing inside the neuron, and you can isolate those very nicely, and each one can be passed through this 10x genomics technology that allows you then to profile thousands of genes within each nucleus. You can then get, I'll show you data here's data from 41,985 nuclei. And I know that some of you have seen plots like this, but I want to make sure I explain what this is for everybody, because this technology is so incredible. So I've been using genomic technology in my research over many years, and it always frustrated me because you do this RNA sequencing, or we used to be microarray, and you take a chunk of tissue and you throw up a bunch of genes there, and you don't really know what any of it means. And it's a mix of all these different cells. But with this technology, you actually can identify the cell type and then ask questions about each individual cell type. How did exercise affect microglia or astrocytes or particular subtype of medium spiny neurons? So what this graph is showing you is each one of these dots, you can see it's made up of these little dots. Each one of these dots is a nucleus. And that nucleus has been profiled for thousands of gene transcripts. Remember, these are unedited because it's the nucleus. And so what's shown here is on this UMAP, they're kind of like data reducing, like principal components, right? Each of these nuclei and the position where it is on this map is dependent on all the genes expressed in that cell. So cells, nuclei that are close to each other on this graph are transcriptionally similar. And cells that are far away on this graph are transcriptionally dissimilar. And what you can see is that it's able to cluster all these nuclei into different types. Many of these are identifying particular cell types. Like, for example, these turn out to be the microglia. So as everybody understand what this is, I've now profiled each individual nucleus. We're looking at 42,000 nuclei here, and all the different cell types. These are medium spiny neurons. I forget what these are. Maybe the oligodendrocytes, try something. What you can do, of course, is then look at different genes across all these different clusters. These are the cluster identity at the bottom here. Notice 2 is right here. And these are just showing, this is a dot plot. So the bigger the dot, the more nuclei in that cluster express that gene. The darker the dot, the higher expression of that gene. So you're looking at 2 different dimensions of expression. And you can see that cluster 2 is expressing a lot of these genes. These genes were chosen because they are markers of microglia. And some of them they're all marked microglia to some degree, but some of these are more specific to macrophages. As you know, microglia are very similar to macrophage. So this allows me to identify 2 clusters, 1 and 14, which look like microglia, and 13 have the microglia markers, but more macrophage markers. So essentially, we found out that this cluster, and these 2, they're all macrophage microglia, but these 2 are actually microglia. And if you look more carefully, and I'm not going to go into the details, there's tools now to ask questions like, what is the cell maturity like? We know that as cells mature, they express certain genes that are common of a more mature cell. Immature cells have many more higher gene expression of other cells. So we can identify that, okay, these were the immature microglia, and these were the mature microglia. The reason why I'm showing you all this is in this data I'm going to show you, we're going to zoom in on the microglia and ask, I'm not going to go over all the different groups, but we're going to ask, are there any genes that change in those microglia that are correlated with running levels? Or are there any genes in that microglia that are altered between the animals that were stressed and not stressed, right? And because the idea here is that maybe some of these, the microglia were altered from the stress, and those microglia then are somehow affecting the striatal circuit and dampening running, or it could be the other way around. So I want to show you this one. Now, what I've done now is basically take this cluster of neurons, and I've reclustered them. So you take them out, right? Because that first map was all in cells, all in the striatum. Now I'm just taking the microglial cells, and I'm asking the program to recluster them. And it does so, and you can see these are animals that are sedentary, both stressed and not stressed. And here's our animals that are, I say exercise enrichment for a reason, and this is directly related to something that Frank was saying at his retirement party, is that sedentary behavior and comparing animal running to sedentary is not the same thing as looking at exercise. There's a huge deprivation when you look at an animal that's isolated in its cage versus an animal that actually can move around. And a lot of that difference, it may have nothing to do with actual running, but just being able to do something, and have some normalcy. So that's why I call this exercise enrichment. And what you can see is there's a whole subtype of microglia that are only found in the microglia in animals that have access to running wheels. This cluster is basically gone here. And if you look at what's different about these genes between the two groups, what's different about this cluster between versus the other clusters, you can see a number of genes that identify certain type of activation. This is not like a canonical type of activation like you may have heard of M1 and M2 type thing. This is a unique flavor of these microglia that are altered in animals that have access to running wheels. And I want to say also, I'll show you in the next slide, you know, there were a couple hundred genes like this that differentiated animals that had access to running wheels versus not. But when you look just at the runners and ask which genes in microglia, remember, which genes have their expression correlated with individual levels of running. Now you find 700 genes, okay? 700 genes in microglia, their expression is strongly correlated with individual levels of running. And those genes don't overlap very much when you compare just the runners versus the sedentary end. So this is another, I think, really reinforcing the idea that when you just compare two groups, runner or enrichment with not runner, you're not necessarily looking at an exercise. You're really looking at something a little bigger than that, more about deprivation, access, enrichment, at least for microglia gene expression. But we see really tremendous correlations between many, many genes and running levels. But think about it like this. Now we have animals that were stressed. We know the stress dramatically reduced running. And we know that that running was also correlated, and stress was correlated with many genes. So we can do a mediation analysis and ask the question, stress reduces running, right? I just showed you that, a massive effect. Well, stress might reduce running via gene expression. That's not unheard of since microglia are inflammatory. They can release factors. Like I said, we're usually thinking of those medial spiny neurons, but no, we should be thinking about the cells that can control many neurons. So we found actually a number of genes through this mediation analysis. And this gene, I don't even know much about these. These are transcription regulators and co-repressors. This is an example of a gene where the mediation analysis said that stress reduces running through regulating this gene. Now, with mediation, you can do the opposite. I would have expected most of the genes in microglia that are strongly correlated with differences in running would just be a reflection of the running. Like if they run more, there may be more activity in the striatum, and then the microglia are showing more gene expression. And we see that as well. This is where stress affects gene expression via running, right? Stress is affecting running, running affects its gene expression. So with this tool, we can actually tease apart. Now, this is mediation analysis, right? So it's not proof, but it's really intriguing evidence that some of these genes may be underlying the actual behavior itself rather than just be a consequence. So the first take-home message I want to give is that support cells in the brain, like microglia, could play a key role in regulating behavior on a longer time scale. We want to understand how genes, how stress, how long levels of running affect the function of neurons. Maybe we should be looking at how the astrocytes and the microglia play. So I want to move topics a little bit here. Actually, I'm just going to run and get my cell phone so I can see what time it is.
Ask questions.
You can ask questions while you're doing it. What's that? Oh, yeah. Oh, yeah, that's right. And when is this over? 5? Okay, so I'm doing a little slow, but that's okay. Yeah. Yeah.
Now, in the genes that you listed there, can you distinguish whether or not they're active or inactive, like ramified or like inactive?
Okay, so we can't obviously tell. Oh, yeah, the question is, so whether you know, so I said that there was not M1 and M2 expression, those genes, but can I say whether they're activated or inactivated? Like, for example, we know in activated microglia, they have a certain morphology where they sort of bulge up. So the answer to that is no, because we didn't do morphology. But my way of thinking about activity is like, well, they're showing all these suites of differential gene expression. And most of them are actually increased expression in actually these stressed animals. So I don't know. I think the field is moving a little away from the M1 and M2 because that sort of started with the macrophages. And now we realize that microglia do amazing things. They're not just sitting there waiting for an infection. They're actively doing stuff. And what this is showing is that they have a very unique profile. I mean, hundreds of genes correlated with individual levels of running. I didn't mention this. It's not acute running, too, because we can look at running right before they were euthanized. What's most strongly correlated with these genes is the cumulative running. So it's a long-term pattern of how much they're running. So whether, I don't know, I can't say that they're morphologically activated, but all I can say is that they show a very unique gene expression profile that seems to be very, very closely tuned to the behavioral state of the animal.
Yeah. For exercise animals or ones that are under stress, if they're in their more active state versus the ramified where they're just resting.
So yes, we do find when they're stressed. We did a score that does M1, M2 by looking at all the genes, and we do see some evidence for. Oh.
So I'm just talking like morphologically, if they're in the ramified or.
Oh, we don't know. I don't know because we never looked.
I didn't know like if those genes are.
Oh, I don't think so.
Ones that have like in a stress situation, I'm guessing that they're going to be more devoid or more active shape.
There were some of them, so that there were like canonical activity, like ApoE was one of the ones that, and that's been found in some of the other models. So there were some evidence for classical activation, but not like fitting perfectly on the M1, M2. So I'm going to move on now because I want to get to some other stuff. But another area that I'm interested in is how does exercise enhance cognition? I spent a lot of time on this. In humans, as you know, there's lots of studies showing that humans have increased volume of hippocampus. This is again done with randomized controlled studies where they have people exercising, 40 minutes a day, 3 times a week for 6 months. They bring them in the lab and they can do quite detailed MRI structural imaging and find that of all the brain areas, hippocampus, particularly the dentate gyrus, seems to enlarge. There's also gray matter changes in the frontal cortex. There's blood flow changes. Optical imaging, I was talking to other people, and this is something I want you to remember later. Hopefully I get to it. I hope I get to it. Is that they've also discovered just these gross features, like the brain of people who exercise are more elastic. So the way that they do this is they use this, my colleagues use this optical imaging tool. This is just showing you some evidence for this. This is epidemiological. So looking at people that have certain cardiorespiratory fitness scores, and as you get lower scores, poorer fitness, you have much stiffer brains. So this is like a macro scale thing. We're not even looking at neurons. The whole brain becomes stiffer. I'm really intrigued. I was talking to people about the blood vessels and the stiffness that occurs there and some of the proteins involved and whether that's happening here as well. So this is happening, but in terms of elasticity and function and mechanical forces and the role that plays on this process is something I want to get to. So remember elasticity till later. And I'm not going to say too much about this because there's been like gazillions of studies, and most of you probably know this, that in animals, they found all kinds of trophic factors, growth factors, dendritic changes, synapses, immune system changes in microglia, astrocytes, another big one I want to touch on, I want to emphasize later. And of course, increased adult hippocampal neurogenesis. This is something that I've been studying intensely for many years. And the reason is I'm interested in this one because it's like, oh, there you go. It's just really robust. It's like you don't need to have a mic, you don't even need a microscope to see the effect. So it's a very robust effect, a physiological effect. It's in the hippocampus. It's very repeatable. So this was done in 2011 with Peter Clark when he was my grad student, looking just across inbred strains, the number of new neurons that you measure there in animals that are sedentary or housed with running wheels, sedentary housed with running wheels. You can see in every single strain, there's a huge increase. This doubles, quadruples. I always like to point out that the CP7 black 6J is the strain we always see, shows like the smallest difference. So a big effect here. The real fundamental question is, how did that happen? How did exercise double, triple, quadruple the number of neurons in your hippocampus? For those students here, you don't realize how profound this is. The hippocampus is the integrity, the cells are deteriorating. 1% a year, my human colleagues tell me, deteriorates after you're 20 years old, okay? But that doesn't mean it's inevitable. You can do things, and the best thing you can do is exercise to increase that. So this is not if we could understand how exercise actually increases the number of neurons that form and revive the integrity of the hippocampus, I hope I impress upon you the impact that would have. And I want to show something that I always showed last time in 2017, and I like to do it every time I give a talk because the exercise brain interactions field is big and growing. And I find that most people I meet aren't even aware of such this important fact about the hippocampus and what's happening in the hippocampus when animals, and probably humans, although it's harder to study, are exercising. And what I'm going to show you here is a picture of the dentate gyrus. This is the part of the hippocampus. Of those of you who don't know, this is such an important area of the brain. This is an area of the brain that is one of the first areas where all your sensory information is integrated and forms unique representations and unique memories, right? My dad was asking me the other day, my dad is 85. He's like, Justin, I remember things from the past really well, but things that happened more recently, like where I went to breakfast yesterday, I just can't remember. Is there a brain area that's involved in that? Well, yes. There's a brain area. The hippocampus, which is critical for forming new memories, is an area that atrophies as we age. And let's see what happens in this area of the brain when animals are actually exercising. So I'm going to show you this picture next, which is a slice of the dentate. You can kind of see it here. It's a really zoomed in picture. It's this crescent shape here. And each of these dots is a nucleus in the dentate gyrus that has a lot of CFOS in it. And CFOS is just a marker of neural activity. I'm not going to go into the details of what it is. We can talk later about it. But you can just see the more dots you see here, the more active this brain was within a 30-minute, 90-minute period before this animal was euthanized. This was an animal that's not running when I euthanized it. And we're looking at a baseline effect of activity. This is what an animal looks like when it's running, okay? And you don't need a microscope. You can just look at the slides and you can tell which animals were running because their eyebrows, which we call the dentate gyrus, are black right there. So huge effect. I'm not the first person to show this. There have been tons of studies where people have recorded from hippocampal cells while animals are running quickly on a treadmill or jumping to different heights. And there's extraordinary evidence that the frequency and the amplitude, the number of neurons that fire and the frequency with which they fire is very closely correlated with the intensity of the movement, the speed of the running or the force that the animal is jumping. And so to me, it's like, duh, this has got to be involved in what's causing neurogenesis. People like Carl Deiseroff have done studies, of course he's the one who did this, but have done studies where he took hippocampal cells and caused little electric potentials in a dish, and he finds it absolutely stimulates stem cells to differentiate and to divide, okay? So if you think about it, it's like a battery. There's like the way this is situated in the brain, there are large numbers of neurons that are firing in synchrony. It's like waves of theta activity you can detect even outside the hippocampus, and it's strongly correlated with running distance. I'm showing you here, and I think I was talking to someone about this, but you can look after day 6, you can look after day 50. The point I'm making with this is that it doesn't matter how long the animals are doing it, they're always going to show an increase in CFOS when they run more. Why? Because this isn't a very acute thing. As soon as the animal starts moving, the hippocampal neurons start firing. In fact, they fire slightly before the animal starts moving. And there's a lot of evidence that these are actually driving some of the activity and are motivationally involved. Of course, it's the limbic area, and this is a little bit of a controversial area because among neuroscientists, most people think the hippocampus is only for learning and memory and spatial navigation, but there's pretty good evidence that it does other things. I'm really interested in the last time I was here in 2017, I was talking about a study to test whether the activity of hippocampal neurons is sufficient to increase neurogenesis. And I came up with this dramatic thing, and it took me like 3 years to do. And so I'm not going to spend much time on it because it didn't work. But I want people to know why, so they don't do it again. So essentially, I have to go through this quickly because I really want to get to some other stuff. But CFOS, I'm basically using this. Animals are running on a wheel. When CFOS is active, I showed you is active when they're running. That basically traps these neurons so that they're forever after going to express channel rhodopsin. So this is a way of me trapping neurons that were activated when the animal is running. And now, for the rest of that life of that neuron, it has channel rhodopsin, which means that I can stimulate those neurons when I shine light on them. And basically, I show this, you can't see this very well, but it's beautiful transfection of the green fluorescent protein in the subset of neurons that were activated. We had these little micro LEDs that we implanted, wireless, it was really fancy. And but the problem is that these wireless LEDs can only activate a tiny little part of the dentate gyrus. And so even though we were able to activate a tiny little piece, and we were able to show, yes, we were able to activate that, that's not what you need to do. You need to activate the entire hippocampus. That thing is like a battery, as I said. There's large numbers of neurons firing there. So what I did here was just was not going to work ever because I was not going to even come close to activating the whole hippocampus. But there was a great paper by Stéphane Remy. Again, nothing to do with brain interactions. He was studying, he's a navigational guy, right? Interested in studying the role of the hippocampus and how animals navigate. But what they did is they stimulate the glutamate afferents that course through the medial septum. And you don't have to detail through the circuit. Just trust me that when you stimulate medial septum with optogenetics and you stimulate these glutamatergic afferents, they cause neural activity in the hippocampus, hippocampal formation. And what I'm going to show you next is from his a paper from him where he basically is stimulating the glutamatergic axons at 3 hertz, 6 hertz, and 9 hertz. And you're going to see what happens 3 hertz, 6 hertz, and 9 hertz, what the animal is doing. And remember, all he's doing is stimulating these glutamatergic afferents, and these are activating the hippocampus at these different frequencies. And 9 hertz is about, on the upper end of the theta, which is similar to level of frequency of firing you'd see in the hippocampus with fast running. This shows you that it looks like that these hippocampal activity not only is just a passive thing taking in sensory information, this is probably driving the exercise to some degree. Okay? And I want to mention something that's really not very popular in my area, because most people in neuroscience or in biology in general don't like side effect hypotheses. They like direct effects. So if exercise is going to increase new neurons, it must have a reason. It must be that those new neurons are important for navigating through the environment or fast speeds to process that. But what about an alternative idea? What if neural activity in the hippocampus is necessary to drive activity, and that you need those activation of the hippocampus so that you can run really quickly? So it's like the step right before the muscles. It's like the intensity generator. And because you're running every day and you need to run more and more every day, like I showed you all them do, you need that activity generator to be a little bit more robust and more cells there to activate the running. Now you have more cells in your hippocampus, not for learning and memory, because you need to run every day. But that hippocampus doesn't care. Hippocampal cells, if they're there, will do other things like spatial learning and other things if they're available. So the cognitive benefits of exercise could be a side effect of the neural control of high intensity physical activity via this hippocampal activation. Testing this is tricky though. And I had one idea where I was going to use and stimulate the glutamatergic afferents, get the animal, the hippocampus to be activated, but paralyze the muscles so that the animal is not actually contracting any muscles, and to see whether that's sufficient. Problem is I haven't really figured out how to mobilize the muscles, but maybe through casting or something like that would be an easier way. But I'll talk to you. I wanted to infuse lidocaine into the spinal cord, but it turns out that's not easy to do in a mouse. So I really wanted to talk about one last thing. Hopefully I have like 10 minutes. Do I have 10 minutes? Okay. I'll try to go through this fast because this, again, is also unpublished data, and it's really some crazy stuff. So I want to, it's very new. We just got a grant from NSF to pursue this with a colleague of mine who's a mechanical engineer. So here is an in vitro model to understand muscle hippocampal cell interactions. So we know the muscles are contracting. We think the muscles are releasing something into the blood. In addition to the neural activity, which again, I think is the primary thing going on, there's also factors in the blood that a lot of people have been studying. And I wouldn't discount that they probably do contribute. So, and we have evidence that they do. So basically what this is, you take primary muscles that you isolate from a mouse, gastrocnemius complex, and you isolate the muscle cell, you put it in this little dish. I don't do any of this. This is my collaborators. And these little, they create these little muscle cells that start twitching spontaneously. You can also stimulate them optogenetically, but they just twitch spontaneously. You can then collect the media from these muscle cells that are contracting in a dish and take that media and put it on a primary hippocampal culture and ask, what does it do to that culture? This is just showing you that when you look at the CM, it has more lactate in it. That's not surprising, but that's just one of probably millions of chemicals that are released into the media. This just showing you the hippocampal culture on these multi-electrode arrays. So they have these electrodes. These are different hippocampal cells, both neurons and astrocytes here. And this will allow you to record from them. And this is an example of the 9 electrodes. And here's regular media, just without any of the muscle contractions there. And you see, you're getting some recording. You can see sometimes the neurons fire together. in synchrony. But this is what happens when you add the conditioned media. Okay, the whole cells fire more quickly. And I was going to show you in the do this. Here is the first is where you take BTS as a chemical that is applied to the muscle media that actually prevents the muscle from contracting. Because one thing you could say is, well, how do you know it's the contraction of the muscles, Rhodes? It could just be that the muscle cell is there. And if the muscle cell is there, you take the media, it doesn't matter if it's doing anything, it'll affect the neurons. Well, that's not true, because when you take BTS, which basically is a chemical that prevents them from contracting, now you no longer see the stimulation in spike rate from conditioned media. This is the conditioned media, the muscle conditioned media, and you wipe it completely away when the muscle's not contracting. So the contracting muscle is important. And from that media, you can see that you get increased neuron number, you get a faster maturation of synapses. The brain network comes together faster and starts firing synchronously faster. But the coolest thing about this is the biggest discovery, the biggest change in that hippocampal primary culture that we observed when you add conditioned media is this massive increase in the number of astrocytes. These things made up basically a layer on the culture, okay? And so we found this really intriguing because, and I spoke about this last time, we had just done a study where we electrically contracted the muscles in an animal that was anesthetized. This is using the sciatic nerve, we stimulated that, contract the muscles repeatedly. And we looked in the hippocampus, not expecting to see anything really, because I didn't think muscle contractions alone could do much. But it turns out that the one thing they did do is increase the number of astrocytes in the hippocampus, just purely contractions. Animal is anesthetized, so there's not a lot of neural activity happening, so there's no neurogenesis or anything, but the muscle media is capable of generating more astrocytes. Astrocytes, I am just so intrigued by now, because especially if we're thinking about how muscle might contribute and affect neural activity. Because look, at astrocytes are situated perfectly where most factors from the blood are not going to get access to the brain until they go through an astrocyte. And so it's not surprising that the astrocytes are one of the first players to both proliferate and support. And the question is, many people who are neuroscientists, they hear me talk about proliferation of astrocytes, they get really upset. They're like, Justin, that's terrible. Astrocytes, that's damage to the brain, and astrocytes are part of that's what happens with Alzheimer's disease. Well, that's true, but with exercise, astrocytes increase in the dentate gyrus to help support the neurons. And astrocytes play a critical role in helping neurons differentiate and integrate. And so I think that the increase in astrocytes that we observe in culture and in vivo are probably functionally important in terms of the exercise response. And here's where it gets real crazy. And I'll just do a few more minutes and I'll stop. But Tahir Saif has spent his whole career, he's a mechanical engineer, and he studied contractile properties of cells. He had a published paper in PNAS many years ago where he took a Drosophila larvae neuron and he cut the axon. He cut the axon, and what he sees is it springs back like a piano wire, right? These things are under tension. And not only that, when you cut the axon and it springs back, all the vesicles that were at the synapse move away from the synapse. And now you grab the axon with little tweezers and pull it, and you find all the vesicles go back to the synapse. So this is the first evidence that tension of these cells is functionally important, right? Now, it's not surprising because remember that the filamentous actin all here. These vesicles have to traffic along these filaments. And if you think about the filaments all folding on each other, then there's nowhere for them to move. They need the Rhodes to be straight, right? And so the amazing thing is brain cells, including astrocytes and neurons, are under tension. The question is, first of all, does the contracting muscles that are contracting release something that might affect contraction of brain cells? Could it be that stupid? Or could they play any functional role? Well, this is the brilliance of a graduate student, Joy, who's working with Tahir Saif, who basically has this 3D culture where he allows to plate primary neurons and astrocytes, and they anchor themselves on either side of this apparatus, and they extend processes and form a network here, anchoring on either side. And so when they form these synapses and connect either side, it can pull the two together, and they can measure the force here. And what he discovered was that muscle media causes a massive increase in contractility of hippocampal astrocytes. And this lasts for 24 hours after application, look at the effect here. These are two different cells looking at a regular media, then changing it to the conditioned media from the muscle contraction. You see astrocytes start contracting, and they continue to contract for quite a bit of time. Not only that, when neurons fire, and you probably can deduce this from what I was saying, they contract, because part of that contraction is involved with the movement of the vesicles to the synapse. And so amazingly, when you apply the conditioned media to mixed cultures of neurons and astrocytes, the neurons also contract. That's not surprising, though, because I showed you they were activated. They fire action potentials, and when they're doing that, they're contracting. The really cool thing is when you apply the regular media to a pure neuronal culture, it doesn't cause them to contract. But if you take conditioned media and apply it to mixed neuron and astrocytes, and then take that media and apply it to pure neurons, then you see the neurons contract. What this means is that the astrocytes are absolutely contracting and releasing factors that are causing the neurons to fire or not to fire or modulate. And the last thing I want to say is, does this matter? Does this have any functional relevance? And here's a really cool, here's an astrocyte. And look at this. This is another way of measuring force. There are beads underneath this matrigel. And when that thing pulls, the beads move a little bit. And so this is just showing you the forces that this cell is exerting on the substrate. And you can see it makes perfect sense. You can see the processes here. And what this shows is that if you increase the stiffness of the substrate where these brain cells are sitting on, it increases the contractile force. So the more stiff that there's the media that they're sitting on, the greater forces they're exerting. And so this is a really cool thing. You can now ask, does manipulating the force alone without the media, is that sufficient now to cause some changes in neurogenesis or something, right? Or does the conditioned media actually increase neurogenesis or neural activity through contraction? And with this experiment, you can kind of address that. And we're basically looking at the effect of the force on neurogenesis by looking at DCX. I'll show you DCX and neurons. They both look the same. So I'll show you this last slide here, and then I'll end. But essentially, the answer to that is yes. If you look at here, 1 kpm RM, 5 kpm RM, and 10 kpm RM, as you increase the force of the cells without any conditioned media, you increase the number of DCX positive cells in that media dish. And what's interesting, of course, when you apply conditioned media and you also apply force, you also increase. But notice that if you look back here, you can see that 10 kPa RM is pretty similar to 5 kpm that has, I guess these two are kind of similar. But if you look at these two bars here, you'll see that the CM is higher. So what that means is that CM, the conditioned media, is causing these cells to contract, and that probably is contributing to the new neurons forming, but not the whole story. There's something else as well. And so really interesting stuff. Take-home messages here are astrocytes are key brain cells at the interface of communication between neurons and muscle cells. And I'm really a big fan of this. I think there's a lot more to do here. Muscle cells release factors that cause astrocytes to contract. Who would have thought? Proliferate, and they release factors that support the development of neuronal networks. So final major take-home messages. I think everyone would agree with me. Exercise is crucial for cognitive health. And Frank has done a lot of work in this area. I think we'd also agree that finding the origins holds great promise for recapitulating the benefits, especially the hippocampus and the integrity of the hippocampus. There's likely some stuff in the blood. I mention this only because I have another whole project, and I wanted to mention it because I didn't realize you did the work with NASA, but I now have a grant with NASA with Marty Bopart where we're doing the hind limb suspension, and we're taking extracellular vesicles isolated from exercising humans and exercising mice and injecting them into the sedentary mice or the mice that are immobilized to try to see, well, one of the group is trying to see out what this does to the muscle, but what we're doing is looking at the brain. And I can tell you that it may have an effect, but it's going to be a small effect in the brain, if it's anything. But there's something in the blood, obviously, from the conditioned media that I showed you that's doing something, I think, particularly to astrocytes. The contribution of hippocampal activity needs to be investigated. I really think that's a huge factor here, probably the biggest factor, but really hard to study and difficult to prove. And lastly, I want to say that support cells like microglia and astrocytes, who when I was learning neuroscientists, no one even cared about. but these are really, really important cells, I think. And the reason why they're so important is because I think they make it really tractable and understandable how something like genes or stress or levels of running, chronic exercise that happens over longer timescales can affect short timescales of neurons. And from that, I will thank everybody. There are a lot of people involved in this because this is a lot of collaborative research. Here's Joy, who did all that great in vitro stuff, building these 3D force sensing things. Ki-Yoon did a lot of the other work with in vitro. Jenny Gardner did the work where I stimulated the sciatic nerve, and I was looking at astrocytes there. Here's Peter Clark, the way he currently looks, and Zach Johnson, assistant professor at Emory University and at Iowa State. And here, Marnie Bopart is my collaborator working with the NASA project. And Tahir Saif is a mechanical engineer. And other than that, this is what my lab looks like. This is Megan Connolly, who's my graduate student now, who's working on the exercise brain stuff. I also have a completely different project that I won't bore you with, but some of these people here are also working on a different project on clownfish. If you're interested in clownfish, we can talk after this about that. I don't think I have time for that now. But I have a few minutes, so I'm going to stop and ask. I'll open up for questions. I don't know. We don't know. So it's like, what's the magic? What's in their magic stuff? I can tell you this. One thing we did is I had a collaboration with Jonathan Sweedler, who's expert at mass spec and measuring molecules. And what we did was we took that conditioned media, and I was like, okay, I asked him, I was like, what's in this, Jonathan? Tell me what's in it. What's the magic stuff? He was like, Justin, I can't do that. There are all these different size molecules. You got to partition the molecules into these different categories. So we divided them up into different size molecules, and we applied each one of those separate size molecules to see if we could recapitulate the effect. And none of those separate samples were able to recapitulate the effect. To me, what that means is probably a lot of shit, a lot of stuff, not just one molecule. And I know there are a lot of papers out there who say selenium is it, or irisin will solve all your problems. That's not been my experience. And most likely there are many, many, many factors, and not even only from the blood. The blood plays, I think, an interesting role in stimulating the astrocytes in some way that probably works in concert with neural activity. But it's a great question. It's the question that I'd love to be able to answer, because imagine if we were able to figure that out and you can take injections. I mean, that was the vision of the extracellular vesicles. They hold lots of molecules in there. So the hope was that they could recapitulate some of the, these benefits in the brain. I'm a little suspicious that it won't have much effect. Great question. Unfortunately, do not know. Yes.
Are these benefits only isolated to cardiovascular benefits?
Yeah, I get that question a lot, and it's a really good question. The problem is that most of the data we have in animals is aerobic. I mean, Frank actually showed I didn't realize that. Did you the one who started the ladder thing? There were some people before you, but there are other ways of doing. But the problem is I can't get mice to lift weights very well, and so human studies are the way to go there, and they're accumulating. Of course, human studies are a little bit. You can't study neurogenesis and things like that. I think what I wanted to do, and I was talking to some other people here, is look at what happens if I really want to know if the hippocampus is activated the same way in humans as it is in mice when they exercise intensely, either biking, which is cardiovascular, or just strength training. My hypothesis is that it's for both. That anytime you need intense muscular contractions of large muscles. And as we were talking earlier, it's not just if I do this in my hand really quickly, there's no hippocampus doesn't care. Okay, if I bred mice for high activity, and when they're moving around their cage, you can see strong correlations between hippocampal activity and their activity. But if you look at normal mice, they walk around their cage, they don't run, you see nothing. So the first thing I'd say is that I think the hippocampus is not going to be involved unless it's an intense activity. And everybody knows you can lesion the entire hippocampus and the animal can still move around. HM could still walk around, right? But HM was lazy, okay? And he couldn't walk quickly. And mice who have their hippocampus removed, they cannot run quickly, they cannot jump far. So I don't know for sure because we don't have all the data there, but there's an increasing number of evidence that strength training as well can produce some of these cognitive benefits. And I think a really good intermediate way to study that is to get someone on the scanner and isolate their head so they can do exercise and just see whether lifting heavy weights isometrically activates the hippocampus. I bet you it does. And if it does, I would be bet millions of dollars that it would increase neurogenesis. Yeah.
The beginning of exercise, person been out for 2 hours and you can see the end of the bridge and you volitionally decide that you are going to push really hard. Right? Is that a different activity going on? That is a
okay, this is a really, yeah. Right. I've been talking a bit with a bunch of people and actually came up with Ted Garland asked me this question, kind of related, because there's a lot of interest here in understanding sort of different areas of the brain involved in motivation, what motivation really is. We think about the nucleus accumbens, right, and the striatum, and those are areas of the brain that are involved in motivating behaviors. But now I'm talking about the hippocampus. What's that doing there? So I think the difference is the striatum, that is involved with like volitional, like if, like you said, you feel like you want to run, or you're habitually running, or you become addicted to it, or something like that. And that's but remember that I could, if I bring in a lion here and it's about to jump on you, you're going to get up and run as quickly as you're possible, and that doesn't need your nucleus accumbens, right? And but if you're going to run, the hippocampus has to be activated. So to me, it's like much closer to the muscle. And I think of it as if you ask a cognitive neuroscientist, where's the intensity generator in the brain? What tells the motor cortex how quickly to fire? Well, it's not surprising that the answer to that is probably in the limbic system, the emotional center. So I think the accumbens, there's different levels of it, right? And the hippocampus is, it's closer that in order to get the motor neurons to fire faster, the hippocampus has to fire faster. But it's not the one that's going to make you want to run. It's once you're about to run, it's what has to happen. That's how I think about it. Yeah.
I was just curious if there was a relation with that.
Oh, so they get longer? Yes. Well, they actually, in the dish, when they contract in the dish, they actually get smaller because you can see in that picture them pulling against the media. So they're pulling against it. Now, the real question is, and I was talking about this, and I'd love some ideas here, because this stuff is very compelling in vitro. But does that happen in the brain? And like, I think when thinking about your question, like, are neurons really getting small, like pulling in the brain? You know, and I think it's possible they could. I mean, they're all connected to each other, and there's things moving. You know, whenever I teach about this, you know, we used to teach it, you know, here's the cell, and here's the nucleus, and the Golgi apparatus, and it's all sitting there, and nothing's moving. But of course, that's totally not what's happening. Things are moving and trafficking and things like that. And so, absolutely, I think these cells are physically moving and tugging and pulling. I think when you have a neuron that's firing, I think this is what I'd love to be able to do is somehow image the neuron and see whether it's actually pulling in a living brain. Is it changing? Is the nucleus deforming slightly when it's getting pulled? We don't really know a good way to do this. The best idea that Tahir came up with, which seems really ridiculous to me, is to section the brain into thin sections, and then you make a hole in the brain. And what you find is the brain then, if the brain is not under any tension, you make a hole, the hole just stays with the hole. But if the brain is under tension, the hole changes shape slightly. And from that change in shape, you can infer what the forces were. I don't know if you can, but that's a little bit different from what you're asking. But so, I mean, I don't know. I know that they could, in culture, they definitely contract physically, they move. In the brain, I assume they would, but never measured it. I would love, we need to do that. I don't know how though. Yeah. They noticed that the frequency of firing the neuronal cortex would increase, and it would proceed like milliseconds before the activity. Now, I talked to Stefan Remy, and I said, isn't this incredible? Like, doesn't that mean that this is connected to the motor system? And he was like, yeah, probably. but I don't think there's been a lot of people who've been following up. Remember that Stefan Remy is not in our field. He's really interested in how the hippocampus works in navigation. And that's the real disconnect. This whole literature out there where they're recording neurons and animals are moving and they're discovering this amazing relationship between the speed of movement or the intensity of muscle contractions and neural activity in the hippocampus. They're interpreting that as like, oh, this is keeping track of the speed at which the animal is moving so that they can update the spatial map. Of course they think like that because that's their area. They're trying to apply it to, but then, I don't know. I asked him this and, his reaction was, yeah, that's probably true. But I don't know. There are not that many people, I think, that are connecting the dots between the hippocampus and the motor cortex. It's a great thing to do. I feel like it's got to be like less than a few synapses because it's got to be like really instantaneous.
Okay, let's hold up there. It's a little after the hour. Let's thank Dr. Rhodes. If anybody still has a burning question, we can ask.